So thank you all for coming. It's really nice to see so many faces. It's lovely to see some people, some familiar people and some new people to these kind of training events. Obviously, in the best of all possible worlds, we'd all be together somewhere lovely. I'd bring cake and we'd be able to do this either on a shore and in person. But we are where we are. And the brilliant thing about this is it means that we can get people from Berwick-upon-Tweed to Northumberland here talking about these exciting things, I think, about intertidal ID. So hopefully you're here to learn a little bit about how to identify things on the rocky shore. That's certainly why I hope you're here. And what's going to happen is we've actually split this into 2 sessions, and that's because, I mean, it's an entire degree learning intertidal ID. What we're going to try and do is just have a conversation to try and highlight some key points that might help you moving forwards, particularly if you're using ID books and getting out on the shore for the first time. And then I'll be available if you have questions, and we can always continue the conversations afterwards. So how this is going to work is there's going to be a first session here on Monday. Going to be about 90 minutes. I'm going to talk for maybe 45 minutes to an hour of that, talking about some of the basics of taxonomy, of systematics, and how we classify things, and then starting off talking about seaweeds, which is my personal passion. Which I will talk far more about a bit later on. Now, on Wednesday, the second part of the session, we're going to recap some of the systematics, and then we're going to talk about animal ID. So between them, we should be able to get kind of basics of the things that are on the rocky shore. Obviously, if you can't come to both, that's absolutely fine. You can always catch up on the recordings later or use the PowerPoints. What we're going to do today is have an initial chat, and I'm going to try and find some time to maybe break out into groups to have a look at some pictures, see if we can do some first level ID of some seaweeds. That is not a quiz, it's not a pressure, it's just a chance to maybe apply some of the knowledge we find and ask some of those questions. So the first part will be relatively me-led, but I'm more than happy to have people jump in and ask questions, or if you have knowledge or ways that you do things or identify things, then please do let me know. I think it's much nicer when we can share things as much as possible. So that said, I'm going to kick off. So my name's Hannah. Hopefully you can see the screen, everything okay. I'm an ecologist by training. I'm a lecturer. I'm based at SRUC here in Edinburgh, but I've got quite a strong background in marine ecology. So I spent a lot of time training people how to identify things on the rocky shore and understanding the patterns of distribution on the rocky shore. So how things are moving and changing, the impact of climate change and why that matters for us. So I'm sure you've all got good reasons to be here, and I'd be really interested, and I'd like to come back to it later. So please feel free to pop it in the chat. If you have any background in intertidal ID, perhaps we've got some experts here anyway, and why you're here. So what you'd like to learn, I guess, and I'll try and kind of touch on these things as we go through. So today we're just going to be talking a little bit about the basics of taxonomy. That'll be familiar to you if you've done a biology degree or anything similar kind of training. If you haven't, I'm just going to give you that introduction to try and understand some of the information you'll be given. Then we're going to talk a little bit about species we find in the intertidal and maybe some tips and tricks to identify them to help you when you go out on the shore. Okay, so I want to start off, for those of you, I know there are a couple that were at a training session we did a few weeks ago, which was talking about blue mussels specifically. But you'll recognise this if you were there. We talked about blue mussels, and what I might call a blue mussel, actually across different countries, has loads of very, very different names. And that's because people use them in different ways. So it might be because they're used for food. So we use moule quite a lot if you're a big fan of French restaurants. Who isn't? But also every language has its own way of sorry. I've just done something clever with my thing. Every language has its own way of explaining what they find on the seashore or animals generally, which makes it quite difficult to talk across international boundaries because the words are so different. Of course, it's not just international boundaries. Even just regional variation can make such a huge difference. So this is what I would call a brown crab. Some people call it an edible crab, which makes sense. If you're interested in eating it, then you are in finding it. In Norfolk, it's called a punga crab. I may have pronounced that wrong, so I'm not. If anyone here is from Norfolk originally, you can let me know if I said that wrong. The words are different in Scots. The difference depending on what part of the country you're from, and it's. I mean, I love this. This is a type of geeking out I love to do because I think it reflects the culture and history of our country and our understanding of how we relate to the seashore. So I think it's a fascinating thing to be able to look at, but it's really hugely unhelpful if you're looking at, for example, population shifts across the country, because I have no idea if what I'm calling a brown crab is the same thing that someone else is calling something very different. Now, all of this is really a precursor to saying the names of things are important. And what we're going to be doing throughout this is suggesting that perhaps instead of using common names, while you can use them and they make a lot of sense, what we're going to try and do throughout this is use Latin names, because the Latin names, the scientific names of our species, are universal. They transcend international boundaries. They're the kind of the language of science, and also they allow us to learn a lot more about that species just by knowing the name, because we know where it sits in the kind of wider evolutionary scale of things. So I just want to touch a little bit on where scientific names came from, because I think it tells us a lot about why they're so important. And I want to take you back to the 1800s. So in the 1800s, we didn't have scientific names particularly for things. People were naming things, but they were doing it very badly. So this is the tomato. And in about 1800, this is the Latin name or the actual name of a tomato. Solanium call inum herbaceoe folus pinnatus incisus racemus simplum, which, you know, it says a lot, actually. If you can speak Latin, it tells you a lot about the plant. It tells you about how it branches, and it tells you about the family it comes from. But if you're trying to order it in the 1800s equivalent of a garden centre, it'll take you a really long time to do so. So this kind of shotgun approach to naming things, by shoving everything to describe it in the name, causes huge problems, not only because conversations become really, really long if you're talking about species. So this is why the system changed. It was changed by this chap, Carl Linnaeus. I'm interested, is that a name that's familiar to most people? Linnaeus? Heard? Yeah, okay, perfect. He's probably one of the most famous scientists, certainly one of the most famous biologists. And that is because he was actually excellent. He was a brilliant, brilliant man. He was a botanist, a zoologist, a taxonomist. He was a physician as well. He was a doctor. So a man of many, many talents. But one of his talents was being really, really annoyed by the current state of the naming systems in the 1800s. In particular, he was a botanist and he hated having to use 7, 8 to 12 words to describe everything because it made no sense, and certainly made learning very difficult. So just be grateful we're doing it today and not at this kind of time. So Linnaeus changed the system. He changed the system of biology by inventing what we now call taxonomy. So how we systematically identify and sort things into different groups. He became one of the most acclaimed scientists in Europe, and that's only because he invented a much more sensible naming system, the modern system we use today of naming organisms that we call binomial nomenclature, which is fun to say as well. All that means it's two-part names, basically, binomial nomenclature, and it means that we give every species a two-part name, one of which is the genus, and the second part of which is the species. So for humans, that would be Homo sapiens, something I think most people will recognize. So this system was started by Linnaeus. He printed what's called the Systema Naturae, the idea of that we should break things down into taxonomy, in about the 1730s. And at that point, it was 12 pages. He wrote down some plants and suggested this is how we name them. 20 years later, everyone sent him samples and suggestions and names, and the kind of 12th edition classified about 8,000 species of plants and over 4,000 species of animals. So it's a very, very different, it changed the ballgame, really. So what does that look like today? Again, I'm going to come back to the blue mussel. What you might see quite often is something like this. The scientific name is Mytilus edulis, and there'll be an L at the end. So Mytilus is the genus, Edulis is the species, and then that L at the end tells you that the first person to name it was Linnaeus. So he was the first person to describe this taxonomically. We call him the authority. A slightly longer way of writing this is exactly the same information, Mytilus edulis, Linnaeus, 1978. That's the first time this species was described, written down, and we call it formally described. So if you're reading biology papers, you'll quite often see the authority written afterwards, because that tells us who was the first person to do this. What you might occasionally see is it written like this. Now, again, if you're reading a scientific paper or a book or something that talks to you about taxonomy, all that means is you write it out in full the first time, Mytilus edulis, and then you can shorten the genus because you've already described what it is. It just makes it easier to write and say. So these are the conventions we'd normally use and things you might see when you're reading through books. It all means the same thing. It just gives you different information to help you identify which species we're talking about. Now, hopefully that makes sense. If you are particularly interested in geeking out about this type of naming, there is actually a full-blown commission whose entire job it is to look at binomial names. It's called the International Commission on Zoological Nomenclature. It has a code. You can look on the website and you can find everything you'd ever need to understand how things are named. It was actually a lovely series of hardback books for many, many years. Now it's a website, which is less exciting, but far more useful. And they do actually meet regularly and make like legal decisions on what things should be called. It's a very, if you're interested in that job, I'm sure it's a very exciting role to take on in the future. Okay, so we have an idea of how to name things. We now need to know what to name. And Linnaeus laid out the same kind of information on this. And basically, in order to differentiate one species from another, he just used what we call morphology, so how they look. That could be the shape, the color, anything that will tell you how one species is different from another. And of course, that makes sense that individuals that look similar are likely to be the same species. But in order to make sure that the name that was ascribed is always ascribed to the same species, we have what's called a type specimen. So that is a single specimen, or sometimes a group, of a species to which the scientific name is formally attached. So that's kind of the anchoring point. If you have any questions about it, you can go back to that type and say, well, the one we described has 5 points on its head, so that's the one this name has. So this is just an example of one. This is I mean, obviously it's not a marine species, it's a butterfly. But what it shows here is that this is the drawing of the what's called the holotype, so the single specimen. And this is the record that everything refers back to. Now, actually, we even still have physical specimens in museums, herbaria across the world. So most species will have that single reference point that you can go back and look at. I would just point you, if you're interested in type specimens, to the Royal Botanic Garden in Edinburgh, because they have a huge herbarium. They have about 3 million specimens in their herbarium. So they have about, I mean, well over half of the world's flowers are examples in RGBE. But in particular, they also have a really good algae collection. So over 100 of the type specimens of algae are available in RGBE. So if you're interested, it's fascinating to look through and see what they have there. And in fact, they're currently digitizing their collections. This is an example. You can look through the RGB websites, you can see all the records, you can see the photos of what they've got there. And if it just enables you to explore a little bit what taxonomy looks like, or did used to look like, and what it looks like today. So I'm not going to go into this in huge detail because, because it's a degree if you want to go into it. But I just wanted to talk a little bit about what a species is. Now, I'm interested if anyone has a definition of a species that you've learned or been told at some point. I can't see you all, by the way, so if anyone would like to unmic and say something, you're very welcome. Looks of fear, mainly. This is not supposed to be a test. I think this is the thing that normally comes up most often for people. It's the kind of thing you learn when you're at school. So you're a species if two of you can interbreed. There's a kind of understanding that a definition of a species. Linnaeus used morphology, so the things that look alike is a species. If you're a paleontologist, you use something very different because it turns out it's very difficult to understand if fossils can interbreed. So you find a way to kind of divide it between the fossil record. They're called chronospecies. It's actually fascinating because you kind of see the evolutionary development through the fossil record. But these days, our main definition, I guess, is from DNA sequencing. And again, from the kind of early taxonomy from Linnaeus, this has completely changed the game. And it's really confusing taxonomists up until this day because it's providing some very different answers. So at the moment, we have about 2 million named species in the world. And estimates of how many more species, distinct species, we could have range from about 5 to 100 million. So really broad estimates of what we don't know. And that's because actually DNA is really changing our understanding of what a species is. I don't want to go into how you genetically differentiate species, but it's important you know that because of our capacity to now rapidly genotype things, in particular things that are called DNA barcoding, so that's taking like short sequences of DNA that are unique to a species and using it to differentiate them, it means that taxonomy is changing. It's just changing so rapidly, no one can keep up. The reason it's important to be aware of this is because if you have a book, an ID book that is 5 years old, some of those species could have changed name. So they might not have changed identification, but it's not meant to confuse you. It's just the whole system of biology keeps on shifting. So it's worth bearing in mind that keeping up to date it can be a full-time job, and just be aware that these things are shifting. If you're particularly interested in DNA barcoding, I'd recommend this. This is a website. This is basically a database of all the DNA barcodes of species we've got at the moment. People are adding to it, open labs are adding to it. So it's worth having a bit of an explore around if you are interested again in finding out more about DNA identification of species. So what has DNA identification told us? It tells us that some things that look morphologically different, or we think are different, are actually genetically quite similar. So in the case of the blue mussel, we have 3 named species: the blue mussel, the Mediterranean mussel, and the bay mussel. Actually, it's uncertain whether these are different species depending on definition. So they can interbreed, but they are in geographically different places, so they don't naturally. Genetically, they're quite different, but they come from a similar evolutionary pathway. So we call this the mitolous-edulous complex. That's in connected species rather than it's just too difficult to understand. But it does mean that it depends on your definition as to whether or not you'd call these a species. And that's not just the case for our marine environment, which is wildly understudied and difficult to understand. Even really super familiar species are now shifting their name. So when I grew up, I knew this blue tit as a Paracerulus, and a few years ago it changed genus completely. So it's a different genus to great tits and coal tits and others. And that's just from a genetic study, an understanding of where this fits in the evolutionary tree. So this is what genetics is showing us, really. This is an example, a representation of what we call the tree of life. So that progression from an early ancestor, the branching of all these different evolutionary pathways to form different species. Now, don't panic. This is not, the details of this aren't relevant. It's just to show you the variety that's out there and the way that genetics is helping us form these shapes. So really, I think taxonomy is frustrating if you're getting into it, but very exciting because it's not just a filing cabinet. It actually tells us this story of the interconnectedness of life on Earth. So when you start understanding taxonomy, it helps you with identification because it helps you understand where things fit. So everything has descended from a single being, essentially. We call it, it's quite nice, we call it Luca, the original single common ancestor, the last universal common ancestor. It's quite nice, it's got a name. All studies show it lived about 4 billion years ago, probably somewhere deep sea vents in the ocean. And after that, evolution just split everything increasingly into different species. Evolved, diverged. And taxonomy is just tracking that divergence of species. I would, if you're interested in tracking evolutionary trees, there's a very nice website. It's called the Open Tree of Life, and it shows you how things connect together. This is, again, a website that shows you how things connect together. It's called One Zoom, and you can track the connections between different species. So it's quite useful, particularly in the marine environment, because what we've often found with people getting started with intertidal ID is that it's quite overwhelming to tell the difference between different things. I remember I ran a training session a few years ago with somebody, and we had a great day, lovely day out on the shore, and we came in and they picked something up on the way back, so just couldn't stop identifying things, so it all got so good. And they picked up and went, look at this, it's a really cool plant, animal, rock. And I was like, that is the classic kind of I'm learning intertidal ID. You can't tell which a plant or animal or rock because everything is so confusing in the intertidal environment. So sometimes being able to explore the way things are related helps you understand where these things sit. I'm going to put this online. There is a brilliant video that takes you through this. So I'd recommend either in the break or after this look, having a look through. I won't show it now. Okay, so why have I been banging on about ID and taxonomy? It's because taxonomy and our kind of classification lets you sort out where things sit. And so it makes ID theoretically easier. The easiest thing to do when you're starting ID is to pick up a book that's a nice book and just flick through and find a picture that looks about right. And that is absolutely fine. That's the way we all start. But the more in depth you get, the easier it becomes to start with somewhere a bit higher up. Where does it sit in this taxonomy? And can I then break it down from there rather than going from the bottom up? And particularly with things like seaweeds that are very complicated, doing that gets you more rapidly to a correct identification. So this is still the kind of taxonomy we use. I mentioned the species and the genus. That's the kind of two-part binomial name we use for things, but everything also has all these different classes behind it. So we start from the whole of life and we break it down to things that are more closely related. And the very top level at that we call domains, or the 3 domains. Now this still feels a little bit new. I don't know if I'm just really old. My very first essay when I was at university undergraduate was basically looking at the 3 domains. So it can't be that new, but it's still not necessarily what we teach children at school. So sometimes this does come as different information for people. So at that very high level of a domain, we break things down into these 3 groups. It's not wildly important, the details of these, but you should be aware of the fact that this exists. So what we call the eukaryotes cover basically everything you would recognize. Plants, animals, fungi, humans, zebras, eagles, everything fit into that one domain. The other 2 are basically single cells. So they're bacteria, anything that is infectious would sit under there. And a very special group called archaea, which are things like that live in deep sea vents and in volcanoes, and these amazing species that only exist in these extreme environments. So when we're looking at seashore, you're never going to be identifying bacteria. You're mainly by looking at things that are in the eukaryotes. And this is the breakdown of what we call the kingdoms. So within the domain of eukaryota, or the eukaryotes, there are theoretically 4 kingdoms. So we can separate things into animals, plants, fungi, or what used to be called protists, which is basically anything else we can't identify as animals, fungi, or plants. I should say that this has all changed. Taxonomists are fighting over this right now. There are so many different groups you won't recognize. For the moment, forget them. I want to acknowledge to you that they exist, but no one has decided how many they are or what they should be called. So for all intents and purposes, they're fighting over a jigsaw puzzle. Don't worry about it. We'll stick with these guys. So in terms of what you're going to find in the marine environment, you will find fungi, but you'll never see them. So marine fungi is a group. It does exist. It's wildly understudied. They're not visible to the naked eye. You don't find the mushrooms that we see in forests. There are about 450 marine fungi species. If you're interested in cultivating seawater and learning about them, that's fantastic. They are brilliant things. They're being used quite a lot for studying how to solve environmental problems. So, for example, after Deepwater Horizon, the oil spill, they found that fungi increased around the oil because they actually can degrade hydrocarbons. So they're being studied to see if we can use them to solve, you know, the Great Pacific Garbage Patch, etc. But in terms of the intertidal, as you're not going to see them, you can always forget about fungi. However, plants you're going to find in the intertidal. We're going to talk about seaweed later, and I'll talk a little bit about where seaweed sits in this. But really, the only true plant, the plant that we recognize you find in the intertidal environment, is seagrass, Zostera marina. This is, I guess, the most familiar species of seagrass. If you're lucky enough to have seagrass in your area, there are about 60 species of seagrasses across the world. Some are more common in the subtidal, some are more common in the intertidal. Out of interest, does anyone have seagrass around them in the intertidal? They don't recognize seagrass. Yeah, a few nods. It is something that it's an amazing habitat builder and carbon store. So a lot of projects at the moment looking to rewild seagrass, which if you're interested, I'd totally recommend following that up. Okay, seagrass is the only main flowering plant. Obviously there are animals, but the one I want to focus on are called protists, and that is that I call it like a rubbish drawer, essentially, of things that we didn't really know where they came from originally. And it was the group that seaweeds have been moving in and out of varyingly for quite a long time. So seaweeds don't sit nicely in the plants. And that's because they are probably ancestors of modern day plants. They don't have the same kind of vascular tissue. They don't have the same kind of root system. And so taxonomy is just moving them around quite a lot. However, like plants, they do contain chlorophyll, they photosynthesize, so they have a lot of shared features. Now, algae is a huge group. There are lots of single-celled algae floating around out there in the ocean, but for the intertidal, all we're worrying about is the kind of what we call macroalgae, so the seaweeds, the big guys that are going to be attached. I think algae is utterly fascinating and well worth learning to ID because, like plants, doesn't run away. And so if you want to sit and look at something in detail for a length of time, I would totally recommend starting with seaweeds. They're also enormously important and I think really underrated in the marine environment. So, I mean, there are various estimates on this, but one suggestion that all algae, including the kind of like floating phytoplankton, are responsible for producing about 90% of the oxygen in the atmosphere. So forget your rainforests, forget the Amazon, forget planting trees, focusing on algae is where it's at. I think some of that comes from how rapidly they're able to grow. So for some of the largest brown algae, these kelps, they can grow about 0.5 meter a day. So yeah, nothing trees. Trees are nothing compared to some of the big algae. What I find interesting about seaweeds as well is they're also have a huge amount of gas exchange. So obviously they're putting oxygen into the atmosphere. But one of the gases they put in is called dimethyl sulfide. I'm not sure, it's called DMS. Anyone recognize that? It actually is responsible for the smell of the sea. So that kind of salty, marine-y scent you get when you go to the ocean and you breathe in, that comes from seaweeds. If you get a little bit of it, it's the smell of the sea. If you get a lot of it, it's that kind of rotten egg, seaweedy smell. But actually, it's almost like a greenhouse gas, because it is released into the atmosphere. It causes water vapor to condense and it forms clouds. So you get more clouds over big seaweed areas. And it actually plays a big role in regulating the Earth's climate. So without seaweed, we'd be nothing. We'd be in huge trouble. Okay, so this is the first step towards identifying seaweeds. They are broken down into 3 groups. All seaweeds contain chlorophyll. That's the chemical that allows them to photosynthesize to get energy from the sun, which gives them characteristic colors. So you get what are called the chlorophyta, the green algae, the rhodophyta, the red algae, and the phaeophyta, the brown algae. Now there was, or there still is, I guess, a theory that the different kind of chlorophylls and colors related to where they sit in the seashore. So theoretically, those that have red chlorophyll are absorbing more blue light. So they sit better further down in the sea because blue light penetrates further. It's got a longer wavelength. And the greens sit a bit further up and the browns sit a bit further up. It's a beautiful theory. It fits really nicely with the physics, and seaweeds haven't heard it, and so they don't really follow it very well. So like pretty much everything in biology, you got the really nice idea and then something will stuff it up by being in the wrong place. But I think there's still a lot of studying to be done in understanding why these use different chlorophylls and why they're different colors. So in terms of seaweed diversity, there's an estimate that there's about 18,000 brown seaweeds, maybe 18,000 green seaweeds across the world, and well over 6,000 red seaweeds. They're the tough group. They're the really diverse group. And although we talk about them like they're similar, they're kind of the same type of group, actually they belong to entirely different groups. So you could argue at the moment, and people do, that the red and green algae should sit in that plant kingdom and the brown algae sit in the protist kingdom. Now that's because brown algae and red algae are about as closely related as brown algae is to me. They are so genetically distinct. They divided evolutionarily such a long time ago that they look similar because they live in the same environment, not because they're evolutionary related. So there's a lot of fights about algae and where they sit and how we classify them, which can cause you some problems if you're trying to look through seaweed books and understand where you're looking. But I think the colors kind of hold true. These are still the main groupings. Obviously, you're going to find kind of reddish, greeny, browny algae, but identifying which of these kind of groups it sits in is going to be your first step. Just take my time. Okay. Now, your next step to identifying seaweeds is looking at the morphology, the shape of them. So if anyone here is a botanist, you'll get very familiar with these. You'll be very familiar with these terms. If you're not, it can seem like a very, very dark and unusual world to go into. But most seaweed books will talk about some of these features. So they'll talk about the holdfast at the bottom. That's the bit that holds it to a rock or to other bits of seaweed, and the shape of that tells you more about it. Instead of stems, they have what are called stipes, so the bits that connect from the rock to the fronds. And then you'll see these fronds that are different shapes. quite often they'll also talk about a midrib, which is this line that tends to go down the middle of the frond. Sorry, I didn't mean to do that too quickly. They will also talk about different types of branching, and it is worth looking at little diagrams on this because it's one of the main ways you can confirm that you're looking at the species you think you're looking at. Whether or not the branches happen on the opposite sides to each other, they alternate along the stipe or the stem, or they do what's called dichotomous branching. That basically means forks. Every junction is a fork. Don't know why it's not just called forked, but it's worthwhile knowing the word because it's going to help you out when you're looking at books. Now, the main useful thing about seaweeds, like everything on a rocky shore, is that it has preferences. And understanding those preferences and looking up those preferences help you identify which species it is. And that's because the shore is very different. If you live at the top of the shore, you have to be very used to being in the open air and very little in the water, and vice versa at the bottom. So everything is adapted to living at different parts. And what you'll often find is something we call zonation. And that is a kind of very recognizable patterns or bands across the shore of different species. I'm pretty sure that's familiar to everybody. Does that look something like you would imagine it looking on your shore? It's not always clear cut. It depends on the shore, but even just understanding where things sit will help you identify them. So let's take a look at this photo and the different shore zones we have here and what species we're likely to find there. This is the top of the shore. So beyond this kind of yellowy white zone, we're out of the intertidal. That is the splash zone. Lichens live there. It's very interesting. I'd recommend studying lichens, but it's not the intertidal. So for your kind of surveys and coastal interests, just step a little bit below that. It's where the seaweeds start that it gets interesting. And that starts with the highest species you normally find is something called Pelvicia canaliculata, or what's otherwise known as channeled rack. This is a very small species. It's kind of bushy and branching. And the interesting thing about it is it rolls over the fronds. So I'm going to spend a lot of time encouraging you to touch seaweeds. Unlike studying lions and bears, I fully encourage you to touch your species because weirdly, the feel of them is often the thing that I use to identify them most strongly. And if you are running your fingers along Pelvicia, or along this channeled rack, you'll feel it kind of rolls into the middle. And that's because it's trying to preserve water because it's at the top of the shore. So it forms these little kind of sausagey channels to hold water in closely. And that tells you not only is it at the top of the shore, but this is the only species that does that. A little bit further down, just below the Pelvicia, below the channel rack, you will find what's called spiral rack, Fucus spiralis. So these are all brown seaweeds. These are the big guys, the ones that are habitat builders and form our rocky shore understanding. Our spiral rack looks a little bit like this. It is a brown seaweed. This is kind of greeny, I know, but go with it. It's a brown seaweed in terms of the grouping. It has a midrib along the fronds that you can see very clearly, and it twists. So this kind of twisting is, again, it's a water conservation measure. A bit like the channels, it'll twist round in order to try and hold it close in. Now, the important thing about this picture I want to point out are these bits here. These are not air bladders. And frustratingly, quite often if you're looking at seaweed books, you will find that it will show these species without these tips on. These are actually fruiting bodies or reproductive organs, and it'll do it at certain points of the year. So some of the year they won't be there, and the rest of the year they will be. This throws most people the first time they're out looking at these species on the shore. Now, the way you can tell that they're not air bladders is they don't have air in them. And again, this is going to come down to me encouraging you to touch everything, in that you can actually run a fingernail or split them apart and you'll feel inside they're kind of gelatinous and spongy. They're not filled with air. So again, just feel them and it'll help you understand what they're for. If you feel that gelatinous stuff inside, it's actually kind of eggs and sperm they're going to release into the environment. Seaweed life cycles are an amazing dark art form, understanding them. Weirdly, the kind of the eggs and sperm you get from spiral rack particularly is a very good moisturizer. If you're interested in that kind of thing, I tend to be able to freak out students, taking them out on the seashore and using it as a moisturizer without telling them what it is. So it's always good for, showing off to at parties, to friends, that kind of thing. Most seaweed facts are, I recommend them. Okay, so a bit further down from your Fucus spiralis, from your Pelvicia zone, from your spiralis zone, you tend to get bladder wrack. Now, this is a species most people are pretty familiar with. It is relatively easy to identify. It's a big brown species and it has paired air bladders, actual air bladders that you can pop. So this is like the bubble wrap rack. And it has the air bladders because it kind of just moves up and down with the water to get some sunlight. Now, the only species you might confuse it with is something that was also found in the midshore. And that is a species called Ascophyllum nodosum. A lot of people call it egg rack or knotted rack. And you'll recognize it because it's got huge long fronds and it's got like air bladders in the middle. So if we look at bladder rack, that has paired air bladders either side of the front. If we look at egg rack, it has one big one that goes along the front in the middle. Those are the main ones that have air bladders. So once you've nailed those, you've got like the main mid part of the shore sorted. So these kind of species, these habitat forming ones, are the ones you should really learn first because they're the biggest contributors to the seaweed on the shore. But also when you're out doing intertidal surveys, knowing what zone you're in tells you what kind of animals you're going to find. So it really helps just to get an understanding of this. Okay. Now, what you may have noticed is that on my picture, I don't have a seaweed band below my Fucus fasciculosus, below my Ascophyllum zone. And that's intentional because sometimes you just don't. In this case, I don't because it's all pebbly and rocky and no seaweed is going to sit there. So theoretically, there would be seaweed there, but the substrate just isn't suitable. So don't be fooled into thinking they have to have every seaweed on every part of the zone. Sometimes it just doesn't exist for various other reasons. So every shore is different. And I would say the more you get to know your shore, the easier it is. So absolutely start locally, start finding out what's on your shore and its kind of quirks and the differences. And then you'll have a better understanding of other shores when you go there. What I would expect to find, if it wasn't such a rubbish shore down the bottom, is something called Fucus serratus. So that is serrated wrack. Now this species, again, it's a big brown seaweed. You can tell it apart because it has serrated edges. That's because it lives in the bottom, it lives in the tide such a lot of the time. It gets pulled around by the water, and so it has serrations to make it more hydrodynamic. So again, you can tell where it's supposed to be because of its adaptations. There's like little clues to everything all the way along the shore. Okay, that's my hint to say that we're going to talk about a few more brown seaweed species. This is how I sort things out here. There are a lot of brown seaweed species, but there are maybe a few more that you're going to find quite frequently that are useful for you to know. So this one is called thongweed, or Hymenthalia elongata. It is quite recognizable because I always think it looks like an Indiana Jones whip. Very long, kind of mucusy, very much the kind of thing you can, like kids like playing with it a lot because you can tie people up with it, things like that. The main thing about this is it has a very distinctive holdfast. So remember I was saying the holdfast is the bit that holds onto the rock. It looks like this. I call them buttons. I don't know if anyone else does, but they're like little kind of duvet buttons that are held onto the shore and the seaweed grows from them. This is just the start of the thongs growing. So if you see those buttons, you know you have this species. No other species has a disc like that. So sometimes it's less about the kind of the wider fronds and the kind of what it looks like, more about these little key features that allow you to identify it. What I would say is that seaweed ID particularly it's like being Sherlock Holmes. There's very rarely a kind of smoking gun that allows you to know what a species is. In this case, there is one, but a lot of them very varied in the way they look, the colour, their kind of branching in different environments. So you're more looking for a variety of different clues to help you identify which species it's most likely to be. So when you start off with these, don't be disheartened by finding a weird bit of something that doesn't match anything in the book. Put it down, move on to something a bit more obvious. Even the greatest seaweed experts I've been out with sometimes have to admit defeat, particularly in the field. Another brown seaweed that probably of quite a lot of interest to some of you is something called Sargassum muticum. So this is also called wireweed. I'm not sure if it's familiar, if you've seen these kind of bladders and branching before. It's actually an invasive species. So quite often it's called Japanese wireweed because it was originally from Japan. And it came to the UK, probably in ballast water. It's a super cool species because it can break off. A bit of it breaks off, it floats around and then just kind of suckers on somewhere new and grows. So it's like a ever perpetuating species. But it started off, it was originally found kind of down here on the Isle of Wight, and it's now up in the Hebrides. That's partly because the UK has currents that go this way around. So everything that starts off down here ends up on the west coast somewhere. But it's an amazing seaweed, but it's very effective. It's very aggressive. It grows to meters and meters long, fill rock pools. So quite a few people are interested in finding that because it can change the dynamics of your shore. It is quite obvious. You have like little kind of tiny air bladders on stalks. No other species has that. And it has kind of, I think they look like holly leaves. They're very kind of scruffy looking and spiky, even though they're soft. So you can spot it quite easily if you have it. Something I'm not really going to go into are the laminaria or the kelps. That's because they're mainly subtidal. They mainly live below the intertidal zone. But if you're doing subtidal surveys, kelp forests are amazing habitats, like the most diverse environments, far more diverse than tropical rainforests in terms of the biodiversity. So if you're interested, happy to talk about kelps, but just to say they're very rarely in the intertidal, so we don't touch on them that much. And then this is kind of the rest of your brown seaweed variety. You can divide them in many ways as you want, but there are loads. One of the ways that some of the really good seaweed books break them up is to how they kind of look and feel. So once you've identified it as a brown seaweed, you can start thinking, is it kind of feathery? Is it got flat fronds? And it'll help you find the right part of the book. Now, what I would say is that there are some species, particularly these small brown species, that you will never identify to species level. It might be that you can only identify them in the lab genetically, or it might be that you really need to be able to take them back and put them under a microscope. I'm going to admit that I am not a taxonomist, I'm a field ecologist. So my theory is, if I can't tell the difference between two species in the field, neither can the animals. And so functionally, it doesn't make a lot of difference. Don't ever be afraid to say, I think I know the genus, I don't know the species, just write that down. It's much better to have an idea of the genus, or even like this is an encrusting brown seaweed, this is a hairy brown seaweed, to get some information than to not have any information because you can't go any further in your ID. That's really important. And always feel free to add notes, and because your information might change over time as you learn more or train more or work with other people. So don't be put off if you can't tell species. I can't either a lot of the time, particularly with some of these. Some of them are easier to tell than others, and we will go through this a little bit later. Some of the species that are easier to spot. Cool, so let's take a moment to say that if you're interested in identifying species of seaweed, I would absolutely recommend stepping away from kind of seashore guides into proper seaweed ID books. I have a lot of seashore books. This is a really nice, I'm not sure if you can see that because mirror imaged, Seashore Safaris. It's a lovely book. It's very well done. It's taxonomically accurate. But the trouble with all these types of books is that they pick out the most likely species that you're going to see. That's fine. I'm pretty much doing the same thing in this session, but it gives you, I think, a false level of confidence that you've identified things correctly. Because if there's only one, if they only show you a picture of one red seaweed, you're like, well, that's it, I've got it, you know. Whereas when you can look at the full diversity of seaweeds, it displays the wide-ranging ignorance we all have about these kind of things. Out of anything, I would recognise this book. I've put it on the PowerPoint. This is a sea search book, Seaweeds of Britain and Ireland, and it has pretty much every species in. It's better to look at the more detailed books and understand that you don't know what the species is than to get a kind of false confidence. The other thing I would really, really recommend when it comes to seaweeds is, if you can, get yourself a hand lens. They're not expensive. And I know that, for example, NatureScot have equipment funds for various different bits of equipment. So if you're interested in getting some for your community, then fantastic. They are great for any kind of botanical ID. And they just let you get that little bit closer to look at some of the smaller seaweeds that might just make the difference for you to be able to tell what a species is. So absolutely recommend those, particularly for green seaweeds. And that's because green seaweeds and red seaweeds are far smaller than the browns on average, and you really want to look closely to understand what they are. So hopefully these are fairly familiar species you might see on your seashore. The ulvas, the ulvaces, most of you probably have these on your seashore in some way. Green seaweeds are interesting. They tend to bloom and fade depending on nutrient load. It can tell you something about the kind of nutrients that are coming on your seashore. I am going to make this a little bit easier actually with the ulvas and say that genetically, it looks like there isn't much difference. So the things that we previously would have called sea lettuce or gutweed, or the names that you might recognize if you look at these, we can't really tell them apart genetically. So feel free to just put ulva and forget about the rest of it. This is one of the few times where you get a bit of a free pass on not being able to identify the species at all. There are around 110 species of green seaweeds in the UK, and the vast majority of them are very difficult to ID to species level. But having green seaweeds there and understanding how much there is does tell you something about the shore. So don't ignore them, just be honest about what is possible. Some other species, green species that you might be able to identify are the codiums. So particularly this one. This is kind of colloquially known as dead man's fingers, or mermaid's fingers, depending on how creepy you want to be about it. And that's because, again, touch your seaweeds, it feels kind of fleshy, but like fleshy that's been underwater for a long time. So it's quite a sinister thing to come upon if you're just running your hands and then find it underneath some other seaweeds. But it is quite easy to identify that kind of velvety, fleshy feel. Another species you'll probably find quite commonly that's green is Cladophora. Some people call it mermaid's tresses. It's that stuff that feels like it's hair underwater, and you'll see it on rocks kind of high mid-shore. Again, it's something you'll probably be able to go out and recognise quite quickly. What I wanted to point out here is that down the bottom, I have put Cladophora sp. If you're not familiar with that, that means I know this is the genus Cladophora. I don't know what species it is. So if that's the case, just write Cladophora sp, short for species. I know the genus, I don't know the species. That's totally fine. Please just go with it. Much better to have that level than to not have it at all. This is the last green one I want to mention. This is Ulothrix, and that is because most of your green seaweeds you are not going to identify very easily in the field. Most of the identification comes from the cellular structure and how they branch. Unless you are super interested in spending time on the seashore looking at cellular structure and branching, don't worry about it. If you are interested and you really want to get a full species list for your seashore, think about getting a microscope. That is something that a lot of people end up getting into. You can take one back again for the community if you really want to ID things to a detailed level. But other than that, in the field, just leave it. Okay, I'm going to finish off with some red species. I should say, in the UK, there are over 350 recorded species of red seaweed. There are loads. What I would recommend is starting off with the ones that are easy to recognize. Record them, and once you've got that in your head or in your skill set, then you can start building and building on it. Otherwise, you'll be totally overwhelmed from the beginning. I also would say, don't be afraid to take photos. Don't be afraid to take pressings of seaweed. You can press seaweeds like you can plants. Build yourself up a little reference collection. If you want to get into this kind of thing, it is a lot of fun. More fun than I'm making it sound, I promise. And so just it's a skill set you just need to build over time. Now, the first one you can probably recognize fairly easily is called Porphyra, otherwise known as lava, if you're Welsh, and like lava breads. It's very edible. A very similar species is actually used for nori sheets, sushi in Japan, or we use the same thing. You'll always find it's like very thin. I always think it looks like, I don't know what's that called? Not crepe paper, like kind of paper you'd use in craft, like crinkly paper, stuck to the seashore. And it's normally stuck to rocks. It's actually a really cool species. It has a totally different life cycle. So this is one stage of its life cycle. The other one looks like a completely different species, to the point where it was only about 1940s that we discovered they were the same species and not two different species. And it was one researcher, one geeky person, who's called Kathleen Mary Drew Baker. And the reason I remember that is because she discovered how to basically cultivate this. She was English, and she gave the information to the Japanese scientists who'd spent ages trying to figure out if they could grow sushi, seaweed, basically, and completely revolutionized the Japanese seaweed and sushi industry, to the point that Japan still has a Kathleen Mary Drew Baker Day every year to celebrate her contributions to their national culture. So seaweed can make you famous if you look hard enough. That was an aside because I was excited. Okay, other species of red seaweed you can probably identify include Corallina. This is amazing stuff. There's nothing else like it. There are a few different species within Corallina as a genus. It is bright pink. So if you spot it in a rock pool, it's pretty, stands out. And it's got a skeleton. So it builds its own skeleton, like a calcareous, like a chalk skeleton, because it stops it being eaten. So it stops all these little munching species. So what you might often find is basically little white seaweed skeletons where the actual species has died, but it's just left that hard shell. So again, if you touch it, you can feel it crunches. Nothing else does that. So it's really easy to spot once you know what it is. Other ones, dulse. Again, if you're into eating seaweeds, I hope some of you are into eating seaweeds. This is one of these superfoods. The idea is, I think, that if you fry it, it tastes like bacon, vegan bacon, just because it's got that smoky flavor. So if you're interested in that, it's well worth looking for. You can find it. It's very flat, kind of hangs on the seashore, and it's quite big. So it's an obvious one. And a lot of people identify it because it's eaten. It is fairly well recognized. Again, basically anything we can eat is more easily identified and more interesting to people. Species called Irish moss or carrageen. If you've ever eaten like a carrageen pudding, it's a thickener, basically. You can boil this up and it acts as a natural thickener. So you still find it in actually lots of different things like toothpastes and moisturizers and vegan gelatin kind of packets this is used. Now, there are actually two species that are very similar. One's called Irish moss and one's called false Irish moss, because they just look the same. So this one here is Irish moss. It's called Chondrus crispus, and it's very kind of flat, dichotomously branched little red seaweed. It grows in little tufts. And so does this one, which is false Irish moss, Mastocarpus stellatus. Now the difference, the main way I can tell them apart, is if you run your fingers along them, the Mastocarpus, the false one, has thickened edges. So the chondrus feels really flat. The Mastocarpus has thickened edges. Again, it's subtle, and you get some looks for feeling seaweed quite often on the shore, but it's the kind of thing that just tells you that slight difference. And the final one I wanted to point out that should hopefully be relatively easy to identify is something called Lithothamnion. Now, it's otherwise known as pink crust, and it's a species that is basically just a crusty layer on top of rock. So it is a seaweed. It looks like a rock. It looks like multicolored rock, in fact, but it is a seaweed species. And it's important to recall because it changes the space available on the seashore for other species. So it's well worth recording for seaweed. Okay, I am aware that that was a wild romp through seaweed ID. Does anyone have any questions or points they want to make at this stage? I can't see everybody, so. No.
There's a question in the chat from Robert, who's curious to know the difference or whether a compound or stereo microscope is best for seaweed work.
I do. I want to study seaweed at this level. I've just used a compound microscope. I don't know what would be best, I must admit, normally, but what I would say is it doesn't have to be super high tech. It just needs to get you down to another level of magnification. So it doesn't need to be anything very expensive or high magnification. Even just bringing it down a level of magnification would make a difference. So in terms of, I guess, moving on quite quickly, if you're interested in studying that, I would say a cheap compound microscope would actually be enough to get you to ID level. Are there any other questions at this stage? What I'm going to suggest, if you're okay, because we've still got another kind of 25 minutes, is that I would like you to have an opportunity to have a look at these things. I would normally do that, obviously, in person. I'd give you the actual seaweeds, but for now, we've got lots of photo records, and it would be interesting if you fancied having a go identifying things. So what I'd like to do is I'd like to, I'll stop sharing this now so I can see everybody. So I'd like to suggest we go into breakout rooms just for 15 minutes, and I'm going to send you a link to a drive that has some photos in it. So I'm not sure if all of you are on laptops or if you've got photos, it might be a bit harder, but I am going to send you a link just now in the chat. So if you can look at it, that would be great. Pop that there. And what that has in it is it has a set of 15 species photos, some of them in the same photo. Have a look. Have a discussion in your breakout rooms. I'll come round them and see if you can identify them. So I've put the PowerPoint in there as well as a PDF. So all the information I just shared is in there as a PDF. I've also put an Excel spreadsheet that says seaweed answers. So you can check your own answers, or if you want to look at the answers and pretend you know what they all are first, that's great too, however you want to have a go at it. But if you're okay with that, what I'll do, I'll break people up just now, introduce each other, have a bit of a conversation, and have a look at those pictures, and then we'll come back again in, say, 15 minutes. So I'm going to just open the rooms now. So you should get a little message up that says, go to room. Yeah, absolutely right. So on the left with the pair bladders for bladderwrack or Fucus vesiculosus, and on the right with the egg rack, the Ascophyllum minutum. Perfect. Okay, easy start. What about this one? Easy start, difficult
No
next step.
Oyster thief.
Oyster thief. Yeah, I'm not. Do you know why it's called oyster thief?
No.
To do with attaching to oysters and then floating to the surface or something in popular mythology.
Really, I genuinely was a question. I had no idea, but I'm gonna take that.
It might not be true, but I think I've read that.
Okay, no, I'll take it as true. I think that's really interesting. Yeah, I guess it's like little air balloons in a way. Yeah, oyster thief, Colpolmenia is the Latin name. And any others in here?
Sea lettuce.
Sea lettuce, yeah. Ulva in this bit. The rest of it, I would say, I wouldn't be putting an ID on that from this photo. I mean, I could hazard a guess, but you know, there's probably a bit of egg rack there. But ultimately, if you're not sure and it's just that battered, just leave it. There'll be other seaweed in other times. Perfect. Okay, species 5.
That is the one that feels like a hair. And again, I can't remember the name.
Yeah, often called mermaid's tresses, Cladophora. That's exactly right. I think this is a great photo because it really looks very hair-like in that photo.
Yeah.
Baby cats in the back.
This one.
Helvetica canaliculata.
Yes, good Latin name. Yes, well done. Yes, Pelvicia canaliculata. Perfect. So the canal is the channel, the canal canaliculata. So you know, some of the names do sometimes help or easy to remember, but yeah, that's not a quick one. Okay, this one.
So the one we thought that the one that is in the background or behind was the Sargassus or
Sargassum muticum.
We went for the knotted rack or the egg rack, large one.
Yeah, difference of opinion on this one. Yes, so this was a bit of a trick photo. So I'm sorry. It is actually egg rack. You're quite right. This is the green bits of egg rack. The bits that do look very much like sargassum in the photo are actually the breeding receptacles of egg rack. So again, another one just to, you know, if you only see one picture of something, very difficult to identify. If you were there, what you'd be able to do is feel that that is not an air bladder, but in fact a breeding receptacle. And this one's interesting, I think, because these breeding receptacles break off and leave tiny holes in the egg rack. And then what grows in it is this species here. So this is actually a red seaweed species. It is what we call epiphytic. So it lives on it like mistletoe. It lives on something, but we don't think it takes any nutrients, just uses it to live on. And it only lives on egg rack. So, I mean, the Latin name is Vertebrata lanosa, but egg rack red seaweed would do. We would know what it is. So yeah, total trick question on species 7 and 8 there, but well done.
And Hannah, can I ask you on that
Yeah.
one? Those the flat sort of breeding receptacles. What time of year are you most likely to see them? I don't know if I've ever seen them on egg rack.
That is a good question. And I don't know off the top of my head. Seaweeds don't have like a spring in the same way. They all breed at different points of the year. And I'm not 100% sure at what point Ascophyllum will do that. It won't do it every year necessarily as well. So you just might not spot it. I will look that up and get back to you then. Okay, species 9.
Corallina.
Corallina, yeah, again, well done. It's that nice pink color. You can see actually the white tips where the calcareous skeleton is. I also think it looks a lot like vertebrae actually along the stipe, but that's something that'd be much easier if you were there in person. You could feel that calcareous. Okay, this one. Any offers?
Dead man's fingers.
Yeah.
Yeah, dead man's fingers, the codium. That's it. You can kind of see the velvetiness and the kind of finger-like qualities. Very, not my favorite. There's 2 species here, 11 and 12.
Yeah, so that was the button in the middle. Was I found where I'd written it down. The Hemothalia elongata.
Yeah,
was the button there in the middle.
Yep, perfect, and well spotted, that button.
And then
yeah,
the other one just with the bladder rack again with the double.
Yep, bladder rack again. This one I think is actually quite challenging because it's very young. So if you look at these little bits here, it hasn't even grown enough to have bladders yet. So once the paired bladders appear, that's definitive. Before that, quite a lot of young species, very difficult. You could probably tell from whereabouts on the shore it was, but again, don't worry if there's a young species in there. If you are doing a survey like the Rocky Shore Quadrat survey, where you're putting a quadrat down and you're only looking in that area, what I would do is, if I only had a tiny bit of this in here, I would just look around me and see what the dominant seaweed species is. If everything else is bladderwrack in that area, it's a reasonable assumption that that's bladderwrack.
I think we did wonder whether it was the spiralis because of having the very distinct midrib. I think that was, we kind of discussed that.
Yeah, it is, and sometimes it can be a little twisty because it's quite young, you know. But if you can't tell, you can put fucus spur if you're not sure, like that's totally fine as well, because again, the balance of whether it's just rock or seaweed species might just make the difference depending on what you're looking at. So good stuff.
You also get, in these more exposed shores, you sometimes get bladderwrack without bladders. Is that correct?
Yes, that is a so quite a lot of the fucoids, the fucus species, the morphology changes if they are in very close to fresh water, or if they're very exposed, like they're being very easily attacked by the tide, essentially, in which case you can get very dried, elongated, bladderless versions. I should also say, in the interest of full information, that Fucus spiralis and Fucus vesiculosis, so spiral and bladderwrack, can interbreed to form some kind of hybrid. So again, if you're really into it and want to look at all this kind of stuff, great. If you just want to write Fucus spur, absolutely fine. The complexities of this can be quite daunting. Cool point though, thank you. Species 13.
Is the Chondrus crispus?
Yes, so I would say I couldn't be certain whether it was the Chondrus crispus or the Mastocarpus stellatus from a photo. You're absolutely right, but I would be uncertain about absolutely nailing it down. Just by looking at it and zooming in, I would say it maybe has thickened edges. So if you forced me, I would probably say Mastocarpus, but I wouldn't be able to tell from a photo. So that is a really difficult one.
Any trick? Sorry, Hannah, I was just saying on that one, because I sometimes find they can be really tricky to tell between the two. And is that another one that kind of, depending on the shore, it's different?
They do have very different morphologies depending on whether they're in rock pools or out of rock pools or different shores, but not in ways that are very easily distinguished, if that makes sense. So the only defining characteristics between the two are, so for mature Chondrus crispus, for Irish moss, if you hold it in a rock pool, it quite often has a purpley violety sheen. It iridesces. Now Mastocarpus doesn't do that. So if you can see the iridescence, that's definitive. Mastocarpus has those thickened edges I was talking about, but that's a bit of a judgment call as to whether or not you can feel that. The only other, I think, definitive difference is that Mastocarpus, when it breeds, has papillae, so little kind of raised spots on the surface. So if you've got little raised spots along the fronds, and you can really feel them, they're like braille almost, then it's definitely Mastocarpus. So those are the only kind of really definitive differences. Otherwise, you're down to judgment call.
So if we were doing one of the surveys and we were doing them, I mean, for when I do the bird surveys, for example, if I see something new or something that's unlikely for the area, you need to give sort of a bit of a description. I mean, is that a thing to, would that be something that you would do, is you would say, oh, well, we felt the raised spots, so it could be a, would you sort of try and put why you've gone for that particular identification?
Absolutely. I think that is great best practice, particularly if you're hoping to build up records over time, because it just allows someone to go back and see if they're would make the same call. So again, anything you can do to aid data verification, whether it's taking a photo, writing down your reasoning for why you've identified it like that, or pressing a sample, any of these would be great ways to kind of just aid the information. Okay, let's do species 14. This is a tough one. Any offers? No. So the main one here is actually Sargassum muticum. It is the wireweed. And I know this is quite a far away photo, but it has a very kind of distinctive wiry appearance, I think, so you can generally tell from far away if you know what you're looking for. I would not expect you to do that without knowing what you're looking for. It's also a species that is in rock pools and fills it quite a lot. So this is a very characteristic kind of pattern you will see. You can just see the little dots, which are the air bladders there. So tough one, but again, it's quite nice sometimes to see things from far away, because I think it helps you get your eye in. And then last one.
That serrated rack.
Yeah, hopefully a slightly easier one to finish. That is serrated rack indeed. Classic serrations, and then those little white hairs, and then some kind of kelp underneath. Not quite sure from this picture.
I've seen examples of the white hair on it, but I was just wondering what they were for.
I mean, that's probably a difficult question with all seaweeds. There is some research that suggests it actually again helps the hydrodynamics. So by disrupting the surface a little bit, it allows the water to flow past rather than tear off. I don't know if that is actually true, but that's the kind of chat. It's a good question. Right, I'm aware we're kind of running over time, so I would like to wrap it up there, but a really amazing job. Obviously, there is quite a lot of expertise in this room already, which is fantastic, but hopefully you had an opportunity to discuss and pick up something from this session, and it would be really nice. Again, I'm always happy to talk more about seaweed, so if you have particular photos or species you're struggling with, please feel free to message me. Really happy to talk about that in the future. I'll put my email in the chat. Obviously this recording will be available online, but are there any other questions, comments at this stage before we just wrap up this evening? No. That's perfect. Sorry, it's been so rushed. It feels like 90 minutes go so quickly when we're talking about that, but I hope you've taken something away. It's been really nice to meet you all, and I'll hopefully see some of you on Wednesday when we can talk about animals. So thank you very much.